glass or fibers on an aluminum or wood support or frame so that this densely pleated fiber of glass and paper including makes is very effective to filter any pathogens. So functions by providing a system of air supply and removal designated to minimizing escape of aerosols from the cabinet. So it could circulate within the cabinet. Standards of HEPA filters based on efficiency of removable particles, zero point 0, zero point 0.3 microns, micro M is called the microns, most typical size of the particle to filter. Filters are easily damaged and must be tested and certified. When installed, when moved, annually at minimum, after many repairs or modifications, it's okay. What you have already know, there are what type of biological safety cabinets are. There are three types: class 1, class 2, class 3. Class 1, air is not recirculated within the cabinet. That is the very basic one. Air is directed away from the operator. Air is HEPA filtered, then discharged to the atmosphere. So it's only the class 1 cabinet is going to has the HEPA filter. But how it's going to taking away anything is very minor. When you know very pathogen is very minor, is very common. So that's why is good so that you are not going to be affected. But when it goes to the atmosphere, it's going to be diluted. It's not that effective or already is in the atmosphere. So air is directed away from the operator. So you have to remember class 1 cabinet only it protects the operator. So air is HEPA filtered, then discharged to the atmosphere. Provides good operator protection but does not protect the product. Example, the cultures from contamination because it's not going away, it's not getting the circulation and purifying. If air leaving the cabinet is filtered but not air coming into the cabinet. So that means not purified air is coming inside the cabinet. It's already is going away from you to the outside. So if there is anything contamination or anything there is a chance if your hand or anything or anyway, the samples can be contaminated. So you doesn't need to know that one. So it's going, coming, moving, taking the air out in the atmosphere. This is the class 1. Class 2, designated for personal, product and environment. Everything is going to protect the product, that is the sample. It's going to be protect you and also the environment. So this is designated for work containing microbes, containment level 2, 3, and 4 laboratories that we have learned what is the containment level 1. So divided into types and subtypes based on construction type, airflow velocities, airflow patterns, and exhaust systems. So basically you have to remember class 2 cabinet is designated to protect personal product environment and what are the containment level used when the pathogens are at the containment level of 2, 3, and 4. So there are different types, type A, type B. Under type A, there is subtype A1, A2, subtype B1, B2. This is the class 2. So these are the different types of filters, what they use and what is the flow of air. You guys doesn't need to know that one. Let me see, 30%, 70% is circulated in the cabinet. You doesn't need to. Just remember that it is this is protects the personal product and environment. And containment level 2, 3, and 4 if the question comes. So these are the pleated filters, how it works on the aluminum, glass, and paper-based filter system, and then how this is, my goodness, we are not engineers, chemical engineers, physical engineers, stupid all the things. So how it works? It is taking the air from here, then going, then circulating, then coming here. From here, it is circulating, coming here. It is going from here, from outside, going, circulating. Then here you can see the holes inside is going to come from purified air is coming. So that's why it's protecting everyone. So working in a class 2 biological safety cabinet when there is a potential for aerosol generation, you must work in the BSc. If there is anything you are vortexing, you are doing culture, you are doing anything. So there is a chance of generation of aerosols. You should work under a BSc. So manipulating human or animal tissue or fluids, opening a closed centrifuge cap after centrifugation, taking spent media off tissue culture, anything, there is a chance. You need to employ good technique. Example, employ good startup procedures. Disinfect everything of you need to work with into the BSc prior to commencing work. Do not block grills. Be seated at the right height. Ensure that nothing you need to use is outside the cabinet. So that means number one, good startup procedures means whatever you need to do that work. First, you have to disinfect with that area. If it is a metal, what is going to use it? The floor? Come on, come on, come on.
What was that?
If it is metal base, the biological subcontinent, what you are going to use to clean?
Oh, not bleach.
2% glutaraldehyde.
Chlorine.
2% glutathione. The
Glutaraldehyde.
sides which are not non-metal, you are going to
Glutaraldehyde.
glutaldehyde percent. Somebody already told the percent. It's not 100%, only 2%. Or the side where there is non-metal, if there is anything, that area you are going to clean it what?
Can use the bleach.
What percent?
1% hypochlorite, 1%.
1% hypochlorite, 2% glutaldehyde, Lewis, be careful. Okay, so 1% bleach, hypochlorite bleach, 2% glutaldehyde, depend upon the surface, whatever you're cleaning. Once clean, at the SOP, you need know what you need. What you need, you need the vortex, you need the inoculation loop, what you need the plates, you need test tube rack, whatever you need to complete that work, you have to keep everything, arrange everything inside the biological safety cabinet. Once you are sitting on the right height, you are not going to, you cannot come out and take another things and then put it, there is a chance of infection. Even you take out the gloves or anything, everything in your gloves container, what you are going to dispose it, that should be in small container, how you work. The glass should be disposed of first inside the biological cabinet, later you can dispose, collect everything and the biohazard bag. Because there you cannot put the whole biohazard bag there, and if it is air or something, there is a chances that there may be contamination. So working in the biological place, everything you need work with inside the cabinet, purge to establish airflow. That means you have to own that thing. Separate clean items from the dirty items. So you are going to keep one side the dirty items and going to do the clean area. Do not pass dirty items over the clean ones. Don't take this one to there. Do not block the front intake grill. So this is called the intake. From here, the actual airflow is coming. Work on plastic backed absorbent material for quick cleanup. So that's why we do that. When it's done, we have the plastic or the something which cannot leak proof. Plastic backed absorbent material for quick cleanup so that you can roll it and throw it in the biohazard, and then you can disinfect. There will be not, if there is any spill or anything, it's not going to contaminate the whole area. So clean to dirty, how you are going to work. So here you all the resources for the cleans, then you are working in the middle, and then see, I told you the garbage bag is also inside. There should be a small sharp container so that if there is sharp, it should go there. And these are all the dirty things, clean to dirty. And this is the class 3 biological cabinets where you are going to work with extremely infectious material, totally enclosed from the in. Here also, you are not going to be, any part of your body is going to be exposed with the things. So you can see this is the hand, it's going to be the hand, is the gloves, it's going to go inside the 1, 2, 3, 4. These are, you are going to put your hands through this one with the gloves. You cannot work, you can only see from here. and we have to work from here. All the samples and other things is going to be arranged from the back side. So it's a totally enclosed gas tight HEPA filter supply, double HEPA filter exhaust. So that is so dangerous. Hard detected, dedicated exterior exhaust, keeping cabinet in negative pressure of 120 PA Pascal. Negative pressure. Attach long sleeve gloves for 4-level pathogens. What are the 4-level pathogens? Example 1,
Ebola.
Ebola. Another one,
Hunter.
That weird one.
Hunter.
Yeah.
Another one,
SARS. Is SARS considered the fourth?
SARS. Yes, very good.
Then there's no fibrovirus or something.
Yeah, that is also, there are so many, but these are very common. Okay, so pressure, though I don't know the Hunter, I know the SARS and Ebola. Okay, pressure gauge on BSc. There is a pressure gauge. You have to see how would you read the rate needle. Figure out the increments like we normally would. 0.75 subtract 0.5 equal to 0.25. So there are 5 spaces between those two numbers. 0 point divided by 5 is equal to 0.5. Okay. So therefore the individual line markings would be 0.5. Okay, 0.5, 0.5, 0.5, 0.5, 0.5. So this is, if it is 0.75, this is going to be 0.7, this is going to be 0.65, then this is going to be 0.6, this is going to be 0.55, and this is going to be 0.5. Okay, so red line is closest to 0.65. Generally, it's the middle. The minimum requirement velocity of our cabinet would be 0.5 meters per second, making 0.65 quite acceptable. So it should be in between 0.5 to 0.65. The 0.65 is the best. This question is not going to come. Maybe it's going to tell you what is the gradation. Generally, it doesn't come. It's a little bit. Spill cleanup in BSc is very important. Leave fan on. You are not going to off the fan because that is the HEPA filter is working. Cover material with paper towels to avoid any splashing. You guys know, I'm just reading it. You guys know everything. We have learned it before. Gently pour disinfectant over the towels, circling from outside to center. That means periphery to center. Then, as usual, wait for 30 minutes. Remove towelettes with forceps to biohazard waste. Then, recover again. One cleaning is not enough. Recover with fresh towels and reflood with disinfectant as noted above, as you did it. Remove towels as noted above, as you did it before. If spilled material goes through the grills, the holes to catch tray beneath, pour enough disinfectant into the catch tray to dilute spill tenfold. So there is the hole, there is the grill. If you see that by chance it has spilled there, you are not going to take out the infected one. So you are going to pour the spill tenfold, the disinfected tenfold, 10 times. If you say, if you feel that there is 1 ml wind inside, how much disinfectant you are going to put?
10?
10 ml. If you say there is 50 ml, how much you are going to put?
60?
No.
If there's 50 and it's 10?
Yep,
That should be 500.
500. It's tenfold,
Damn.
means 10 times. Then let it stand for 30 minutes. Drain tray through the drain cock. There is the opening where we are going to take it out and then clean it again. In biological safety cabinets, there is some UV light. It is strongly discouraged due to limited effectiveness. If used, workers need training in safe working practices related to hazards of UV radiation before they working with it. So generally, you guys doesn't have to UV radiation. If there is a really need the UV, you have to have the special protection from your eyeglass to everything. But sometimes we leave the UV line on at night when there is nobody in the lab, so that everything has to be sterilized or clean or disinfected. Should only be used as secondary method of disinfection. Never rely on a UV radiation alone. Used when MLPs are not in vicinity. That means when there is no one in the vicinity in the lab. Nowadays it doesn't happen because mostly it is 24 hours. But in the research lab tube at the night when there is nobody, we bring the UV light on. But it have to be careful when you are going to on and off the UV light. UV light is, what is the UV length? Can you see? What is the UV length? Is 400 up or down?
Looks like it's down, ma'am.
How you know that?
Because it's already shown, ma'am, on the
On the slide.
slide. I saw it
Uh,
like 200,
Yeah. The one,
250 nanometer.
the one that
how
is
you
up is ultraviolet.
Wha-
Ultraviolet is the
okay,
one
okay,
that
okay.
is
What
above
is the
700.
normal range of the lights? Normal lights, human visible light. What is the UV
400
range?
to 700.
400 to
400 to
700.
700. Yes, ma'am.
Above 700. What is that?
Then that's ultraviolet.
Infrared.
I
The
mean,
Huh?
ultraviolet.
infrared.
Infrared,
Infrared, not
ma'am.
Ultraviolet.
Yes,
Infrared.
ultraviolet is below.
Yeah,
That's
Ultraviolet,
why I
yes,
told
100.
400.
you is if you think the VIBGYOR is starting from the violet going to the red. When there is red is 700, above red is infra. That is to remember. And violet, ultraviolet is below violet. So below 400 it starts the UV. There are two known UV, UVA and UVB. What is the difference of the UVA and what it causes UVA and UVB? No Google search. Anyone knows? UVA is ultraviolet for aging. It works on the cells. So that's why it got the cells become destroyed and cells got age. This damage. And UVB is burned. It burns the skin. So that's why when is the sun ray is too much, you got the burn from the UVB. And even there is a in the daylight, even it's cloudy, anything, there is still UVA light is on. So it's caused the cell damage. So need to routinely test it with the UV meter to verify proper intensity at the appropriate wavelength in the center of the work area. The intensity of the lamp is drastically affected by the accumulation of dust, and dirt on it must be cleaned frequently with disinfectant.
Are we just skipping the first slide or?
Yes, there is no need. I told you.
Okay.
UV germination lamps produce UV light at 250 millimeter wavelength. The intensity is destructive. Capability of decreases by the square distance. You doesn't need it. Exposure is always related to distance, does not penetrate into the cracks through the grills, causes deterioration. This is not important. This is also not important. Nobody is going to ask you the question. But if it is dirty or something, it loses the intensity. UV light should never be on. This is important. UV light should never be on while someone is working in the cabinet. UV light should not be left on while anyone is in that area, which can be likely to be exposed to reflected UV radiation because so it's so hazard. Use only with shielding. You need a special shielding to use special type of goggles to work with the UV light. So generally the lights are like this, kind of ultraviolet. You can see the color is kind of light yellow, light violet. So never touch UV bulb with bare hands because oils may leave a fingerprint and create a dead space on the surface. If you touch this one with a bare hand, if there is oil, the UV light is going to be distracted. That area is not going to, the UV light cannot get out from there. This is the reference questions.
No, ma'am.
Okay. What is next one? These are the different questions. So what is the red line here? 0.65. You guys know that. Okay. Right? This one you understand how to measure it. And each one is 0.5. If they give you some pictures, just give me a second. So if they give you one picture and the red line showed here, what is going to be the measurement here?
0.60.
0.6, 0.6.
Yes, ma'am.
If the
0.6.
red line shows here, they can give you the picture like this and give the red line here, okay?
Yes, ma'am.
Or here. So you have to know that 0.6, each one is 0.5. Okay?
Yeah.
Okay. Got it. Okay. Just give me a second. Okay.
It's 0.65.
This is 0.65. This is 0.7. This is 0.65. I ask you here.
Yeah.
This is going to be 0.6. This is going to be 0.55. And this is 0.5. This is 0.5.
Mm.
So this is the hand washing. Do I have to repeat it? You guys know this one. I'm not going to repeat it. You know this one, right? How to do the hand wash?
Yes,
ma'am.
miss.
When you are going to, you must do the hand wash when it's visibly soiled. Otherwise, you can do the hand hygiene with the sanitizer.
Sanitizer.
Yeah.
Okay? Okay. This one we know how to put on, done and done iffy. So this is the hand sanitizer. How we are going to 70% isopropyl alcohol. Then you know how to rub it. Then you know how to do this one is the alcohol-based sanitizer. Dawn, Dove, you know everything. So I am not going all the things. How to remove which one? You guys have to remember the order of donning and doffing. Okay? I don't need to remember. So how you are going to don in? Just opposite is to doff up. So which one, if you have to wear the mask, gloves and eye shield, what you are going to wear first? Gloves, gown and face shield. Which you are going to wear first?
Put on gown.
Your
The
gown.
Put on
gown.
Gown,
face shield.
Gown,
Then
after
gloves.
gown,
Mask,
then mask, then
mask,
Mask.
then
eyewear,
goggles,
gloves.
then
okay. And when you are going to put up, what is going to go first?
Gloves.
Backwards,
Gloves,
gloves.
then
Gloves.
Protective eyewear, mask,
Then your high wear.
and gown.
okay,
Gown and then
okay,
eyewear. And
okay. So first you are going to do the hand hygiene. So I'm not going to. This is can be done. So next one. Okay, go for break. Then we are going to go. So how many units are there? I don't know how I'm going to complete it. Okay, let us go. Go for break. You need a
Thank you,
break or
ma'am.
no break?
Yes, ma'am, we need the break. Thank you.
Not at all.
Yes.
Yes,
Yes.
yes, ma'am.
Yeah, I'm going for a break. I'm gonna go to the hospital and I'm gonna be on team, but when I get there, I might be off.
Okay, so you are going to the hospital now?
Yeah, I'm gonna go there.
You are feeling bad? You are feeling worse pain?
Yeah.
Okay, go. You will be able to drive, or is there anyone to drive you? Or you are going to call Uber?
No, I can drive.
Okay, be careful. Okay?
Mhm. Okay. Thank
Okay.
you. Okay.
Okay. Alexi, why you are so late today? Okay, it's almost time to come back. It's more than 30 minutes. Please come back, everyone. So we are now starting the real world of microbes. What are the different types of microorganisms? We can see what are the bacteria, what is the viruses, what type different kind of bacteria we have. So these are the different types of bacteria. You guys can see. We are going to describe one by one. What are these? So there are course objectives: identifying bacteria, virus, parasites, protozoa, fungi, chlamydia, and rickettsia. The classification, nomenclature, how they become named from grandfather to the great grandkids. So what is the meaning by normal flora? What is pathogenic organism? We have all learned in before. So, but just to refresh again and including couple of them, little bit details. So what is the role of the microbiology departments? You know what is the prefix micronemes, you already know. So role is to isolate and identify various infectious organisms or pathogens. That is the microbiology department deals with. So basically microorganism cannot be seen by the naked eye. So for seeing this one, what you need them? One scope through which you can see them, that is called microscope. Microscope to visualize or see the microorganism is called the microscope to view the structure. Microbiology is mainly divided into 4 groups: bacteria. The study of bacteria is called bacteriology. Logy is study of bacteria, bacteriology. Viruses, virology. Parasites, parasitology. And fungi is mycology. All can be easy to remember, but fungi is mycology. So myco means fungus and logy, mycology. So this comes there. So make sure you can remember the fungi. Others you can understand from their name, but what is mycology? Study of fungus. Prokaryotic and eukaryotic cells. We have learned when we have learned the cells in the anatomy. All living organisms can be classified according to the cellular makeup, means what is inside the cell. Cells are either prokaryotic or eukaryotic based their biochemical structure, because cells is totally full of biochemical structure to function. Prokaryotes, what are the prokaryotes? Prokaryotes, pro means before it forming a nucleus form. That is called the prokaryotes. The prokaryotes doesn't have a nucleus or any membrane-bound organism like lysosome, mitochondria, different things, Golgi apparatus. Those are the membrane-bound organism, mitochondria or Golgi apparatus. You guys can remember the cell structure. Lysosome. There are individual structure inside a cell. Cell is covered by a membrane. What is the name?
Cytoplasmic membrane.
No, cytoplasmic membrane.
Or the cell membrane.
Cell membrane. What is another name? Plasma membrane.
Protoplasm.
Plasma membrane. Protoplasm is what is the nuclear plasm is a cytosol is present inside the cell.
Okay.
Plasma membrane or blood
Plasma
cell membrane, that is rough at the cell membrane. So a cell is covered by a barrier, it's a cell membrane, and inside the cell there are different organelles which are membrane bound, like where is the Golgi apparatus, what is the endoplasmic reticulum, what is the lysosome, what is the nucleus, what is the what is called mitochondria, anything is individual is surrounded by another membrane inside the cell. But in prokaryotes, they have only the DNA. They doesn't have any nucleus. Where the DNA stays in the eukaryotic, in the main cells, it stays inside the nucleus. Right? In nucleus is a membrane-bound organ which is present inside the cell. Inside the nucleus, there is the DNA. What is DNA?
deoxyribonucleic
Nucleic
acid.
What is that? What is this important? What is that?
It contains the genetic materials.
Genetic material we transfer from one generation to another generation. So it says prokaryotes main thing, it has only the genetic material as DNA. It's typically consists of a single circular chromosome located in a nuclear region within the cell. You doesn't need to know all those things. Just remember prokaryote has only the DNA and doesn't have any membrane bound organelle. Okay. These organisms have a cell wall that contains peptidoglycan that you are going to see, peptidoglycan layer, you are going to later see. This presence and absence gives you the gram positive and negative stain. So you have to remember the peptidoglycan layer. We are going to come later when in the staining. What is that? A protein and carbohydrate sector. Peptido means peptine from protein and glycan is from carbohydrate. So it's a combination structure of protein and carbohydrate that aids in maintaining cell shapes and prevent osmotic lysis. What is osmotic lysis? The difference in the concentration of the solution where they're living, in the environment where they're living. So they can protect themselves. This peptidoglycan layer give them strength for any osmotic lysis or difference in the concentration of the inside and outside of the fluid. Bacteria are prokaryotic organism. So bacteria, this is another important. Bacteria is prokaryotic or eukaryotic organism. Bacteria is prokaryotic organism. So that you have to remember. So prokaryotes, they have, see, this is the bacteria. There is DNA, it's a circular DNA. And there are some other structures, but nothing is membrane bound. Not inside. When you are going to see eukaryote cell, then you are going to see the ribosome is a complex molecular machine found within all living cells that serves as the site of biological protein synthesis. It needs to have a protein synthesis so that it can survive, it can multiply. Eukaryotes. Eukaryotes means true nucleus. Pro is before nucleus, eu means true nucleus. An organism composed of eukaryotic cells include animals, plants, and fungi. Eukaryotic cells contain a membrane-bound nucleus, what you have seen before. Organelles such as mitochondria, lysosome, cytoskeleton, cell membrane, and large ribosome. This DNA is enclosed within a nucleus, what I just told you before. It is a typically arranged multiple copies of chromosomes because DNA is ultimately wrapped and called into form with association with the protein. Histone is called the chromosome and is packaged or organized by nuclear proteins called histones. Just I told histones. Histone protein along with the DNA, it make a coil and is called the chromosome. The complexity of eukaryotic cells improves the efficiency for metabolic processes, allow the cells to grow larger, providing for tissue development, multicellular structure. In medical microbiology, the eukaryotes includes the parasites and fungi. We are in eukaryote or prokaryote.
Eukaryote.
Any animal beings, including the plant, fungi, only in our medical terminology, what we deal with the parasites and the fungi. is kind of medical. We are not the medical pathogen, right? Until we transmit disease, but we are not the dreadful pathogen. From us, it can transmit from one to another if we don't do the good practice, hygiene practice, or the preventive measures. So we are required. So basically, in our things, as per the microbiology, and especially we are focused on the pathogens, we have parasites and fungi. So bacteria is prokaryote, parasites are fungi are eukaryotes. That is the main 2 points from these 2 slides. So eukaryotes, you see the structure. This is the cell membrane. That is called the membrane bound. This is the mitochondria, is a membrane bound. Is the vacuole. This is chloroplast. This is the plant cell. That's why it says the chloroplast. You have different endoplasmic reticulum. You have the nucleus. This is the, this is the nucleolus, which is inside the nucleus. Here is the nucleus. Here you have the DNA, chromosome, everything. So every organelle, this is called the cell, and inside the cell, the small one is called the organelle or subcellular particles. Subcellular particles are membrane bound, so that is surrounded by the membrane. Classification, how you do the nomenclature of a microorganism is called the classification of the microorganism. What it gives, it provides an orderly method for placing organisms into categories from where it comes and where it goes. Historically used to be placed into categories by shape and biochemical properties. Due to molecular testings, this is called the genetic testing, we can be more detailed and accurate in our classification system. So basically broad classification, then ultimately you are going to genetic testing to give more specific one. Is used as a standard method for developing nomenclature for living things. Is the method standardized? It should be same, use the same method for nomenclature for different types of animals or pathogens or whatever. Provides a mechanism of identification of living things. Say, if I say the plasmodium, it can say which kingdom it is, I mean, from which country it is. Say, for example, you will see that particular groups. So, types of groups in microbiology, as we told, we have bacteria, virus, fungi, and mold and parasites. HIV is virus or a bacteria?
Virus.
Virus.
Yeast,
Fungus.
Fungus.
plasmodium,
Parasite.
That's my parasite. Malaria parasite.
TB.
Bacteria.
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 Okay,
Bacteria.
good, good. So there are some couple of examples of bacteria, whatever you already know. In addition, MRSA. This is, they are the resistant. MRSA. Staphylococcus. This is bacteria. COVID-19, you all know, is virus. Influenza is a very common virus. HIV, hepatitis B, these are all virus, but fungi and mold are candida. You can see the vaginal, that yeast infection in especially the vaginal regions is the candida. Aspergillus is another. This is causes some fatal diseases. Some parasites, malaria, tapeworms, giardia, lambidia. The dengue is virus or bacteria?
Virus, ma'am. Oh no, ma'am. Parasite. The vector is mosquito. It's a parasite born.
So now classification, how it classification? All living things are classified is taxonomic grouping. It's called the taxonomy and hierarchy nomenclature. It starts with the broadest relationship and progress to the members with the closest relation. Say for example, it started with the kingdom. This arrangement is important, you have to remember. They are going to scramble the order and will tell you which is the right one. Kingdom to phylum, then to class, then what is order, then what is family, and this genus and species. So there are, I think, 8 steps: kingdom, 2, 3, 4, 5, 6, 7, 1, 2, 3, 4, 5, 7 steps. Sorry, 7 steps. Kingdom and then the species. So kingdom, which kingdom is come, which phylum, then class, order, family, genus, species. This is the taxonomy. So the 5 kingdoms within the classification schemes are Animalia, which is animal, Plantae, plant, Protista, fungi, and Monera. This is also called the prokaryote, that is the bacteria. So bacteria is the go under the Monera. So this remember that bacteria is a prokaryote and is go under the kingdom Monera. Okay. Okay. So it is already wrote. I already told you. I am stupid. So what kingdom do you think the bacteria belong? It's Monera. So bacteria belongs to Monera kingdom. Parasites, Protista is parasite. You can see the parasite from the parasite. Parasites belongs to Protista kingdom. So if you go east and molds bring to fungi kingdom. There is a fungi, right? A fungi kingdom or fungi or fungi. What you say? I don't know. You can say anything. Yeast and molds belong to fungi kingdom. Bacteria, virus, viruses. What is the difference between a virus and bacteria? Bacteria are single cells that can survive on their own inside or outside of the body. So viruses are single cells that can survive on their own inside or outside the body. Whether so they are not dependent on the host only. They can survive inside. Either they can form the spore and they can live for years and years after years and after years, or they can go inside of the body and use the host machinery and multiply and cause the symptoms or the disease. Virus can cause infection only by entering the body and multiplied inside the human's host cell cells. So virus always need a host to multiplication. So bacteria, versus bacteria can survive without a host. Virus will die without a host. If there is no host, it's going to die. Free living cells can live inside or outside the body, non-living collection of molecules that need a host. Reproduce by binary fission, reproduce by electric fission, though doesn't need it. It's both humoral, phagocytes, B cells, and cellular T cells. Immune response help in that. They are attack the bacteria. And only the T cell lymphocytes, they only attack the viruses. You has learned, you doesn't have to know CD4, CD8, but you can only know the T lymphocyte are responsible mainly to attack the virus that you have already learned several times. But the B cells and T cells, they are all responsible for the immune response to attack the bacteria. Bacteria is little bit bigger in size. The smallest one is 0.4 microns, where the viruses are much smaller. See, it can start from 0.02 to 0.25 microns. See, it's almost, even the largest one is almost half of the bacteria size. Antibiotics treat bacterial infection. Here the question may come, did the antibiotics can treat virus? No. That's why when there is a viral infection, doctor suggests they doesn't give any antibiotics. Antibiotics cannot kill the virus. Only the antiviral medicines can destroy viral, but which is not effective on bacteria also. So identification, biochemical testing, and PCR testing. And identification by the viral cell lines or PCR. PCR can be tested for both bacteria or viruses. Remember, not all bacteria or viruses are pathogenic. Some bacteria is called the helpful bacteria, which can help regulate our body functions such as digestion. Not
Ma'am,
all, yes,
is that also including the vagina with good and bad bacteria?
it depends. Yes.
Okay.
Yes. But not all viruses are able to replicate using human host and are not infections. Some can develop, evolve, and jump from one animal host to one human host. That's okay. You doesn't have to remember. Only can remember some. We have some beneficial bacteria in your body. Mainly is helping the digestion. Probiotics. What is the probiotics? When there is no probiotics, we feel problem in the digestion. So probiotics, the different collection of bacteria which helps in the digestion. Other microorganisms are protozoa, which is the unicellular element, chlamydia, and rickettsia. We are going to learn a little bit more about that. So we have already learned all known organisms are designated to the minimum of binomial names. You cannot say all names from the kingdom to the species, but generally they have two names they have to give it. One is the gender and one is the species. Gender is little bit broader and species is more specific name. Anyone know what is the scientific name of the human beings?
Homo sapiens.
Homo sapiens. Which
Yeah.
one is the gender or which one is the species?
Species is
Homo
sapiens, I
is,
think.
yeah, Homo
Species is
is the
sapiens and Homo
gender.
is the genus. Yes. So
Genus.
names are generally used by the Latin or Greek words and is always in italics. Genus name is always capitalized, started with, but the species name always start with the small letter. This is question is going to come. They will giving different options to make sure whether you know which one is the right one, the nomenclature. So the first genus name is always start with the capitalized letter, while the species name is start with the lowercase. Okay, and all is has to be italics. And generally they didn't show it here. So whenever you have to write, you have to underline this one and this one separate. That is the actual rule. They didn't tell you that one. So when discussing a genus without a reference to a particular species, the genus can follow with the abbreviation SP, means species. If you doesn't know the name, what is the specific one, species, you can say Staphylococcus species. That means it belongs to Staphylococcus group. Okay, so SP means that abbreviation is the Staphylococcus type of groups. It has the Staphylococcus type of all characters or similarities. When there is a non-formal reference to all members of the group, no capital or italics are used. When there is Staphylococci, so this is the Staphylococci means this type is a group. This is a different types. It's a non-formal, like we are calling the nickname. That is kind of like that. Species. Remember, the species represent organism with the closest physiologic and genetic relationship. The species is more specific. So all species or all Homo sapiens are genetically, individual genetic structure function is different, but in general, as per the human beings, the species is more genetically related. So it is the basic unit of biological world. Depending on the species, like microorganism may cause a different disease in humans and may be apparent in naming of the two organisms. So Staphylococcus epidermidis versus Staphylococcus aureus. From here, epidermidis, you can understand, is going to cause some infection on the skin, right? So now let us go to the different types of microorganism. First of microorganism. Microorganisms means which is naked eye, which cannot see. Be made me pathogenic. It may not be pathogenic. Not all microorganisms are pathogenic. There are some microorganisms which is present all over, always in our body, all over there. So these are the normal flora. It's called the normal microbiota. The microorganism present under normal condition in specific site or human body is called the normal flora or normal microbiota or colonizer. The constituents of human were intestinal tract, especially the large intestine and colon. These microorganisms benefit from this association because they derive essential food materials from host. So what is the symbiosis? We are helping each other. These microorganisms using our host resources to make them alive and reproduce. At the same time as they are taking the benefits from us, they are also giving us some benefits. They are helping in the digestion of vitamins, which is essential for human life. This process is called the symbiosis, helping each other. This is also very important. Another one question, which areas of the body fluid should be sterile, should not contaminate anything? Blood, until there is infection. Urinary bladder, urine is highly sterile. Lower respiratory, that is the pleural fluid, from where there we take out the fluid. If you have to, when there is a problem in the edema in the lung or something, they took out the fluid from the lung, right? And obviously the CSF. So blood, CSF, lower respiratory tract fluid and urinary blood fluid, all these four fluids are extremely sterile. Urine, when it comes out from your urinary opening, then or the meatus is, then it become non-sterile. Before that, it is up to the intensing sphincter muscle, the urine is totally sterile. So in general, blood sterile, CSF sterile, urine normally sterile. Sometimes it may con some E. coli, that is from the urinary tract. What you are telling is E. coli, UTI is not, is doesn't got any UTI. Skin, you can have the Staphylococcus species, Coagulus diabetic. So any type of Staphylococcus which is not causing any harm can stay in the skin. What is the methicillin resistant Staphylococcus? The examples of common, this is the name. Intestine, you have E. coli, you have Pseudomonas sp, you have Proteus sp. What is the example? Salmonella, Shigella. Common pathogen, these are the pathogen. MRSA. So methicillin resistant Staphylococcus. When these are present, they become infectious. But these are the normal ones. So that's, they are telling this is the normal one, but this is the, in blood, there should not be any, it can be pathogen, anything can be go and do the infection in the blood. Same thing for the CSF. Urine normally sterile, but sometimes E. coli can cause some infection. Skin, Staphylococcus species can stay as a normal, and this is MRSA. Is a common pathogen. Intestine, E. coli, Pseudomonas, Proteus. These are useful bacteria, but when you got the Salmonella, Shigella, it causes food poisoning. Throat, strep, viridans, diphtheroids, strep group C, D, E, F, and G. You doesn't remember all those things. So these are stays in the throat. But when it's pyogenes, pyo means past forming bacteria, it's called the strep pyogenes or beta hemolytic streptococci. This, when this comes, is cause the throat sore. Okay, so beta hemolytic streptococcus or strep pyogenes. Pyo means pus formation, genesis. Genes means genesis, formation of pus. Urinogenital, here lactobacillus. They are the common bacteria which cannot cause any UTI and they can stay here, lactobacillus. But Neisseria gonorrhea is another one which is the common pathogen. Pathogenic microorganisms are generally beneficial and essential for life sometimes and sometimes harmful to the host. When microorganisms,
Yes.
hello? Hello?
Oh, sorry,
You
Miss.
got,
No, I have my mic open. I was just talking to myself there. My bad.
okay. When any microorganism, the microorganism which cause disease referred to as pathogen. So again, all microorganisms are not pathogen. Some of them may be normal flora, some of them may be pathogen. Only which cause disease, those are the pathogen. The process of the ability of the causing the disease or pathogenicity. So pathogenicity is the ability or the process to cause a disease by the pathogens or the microorganisms which can cause the disease. It is important to be able to distinguish normal flora from pathogens in a specimen. So pathogenic organism, obviously you guys know it can include bacteria, fungi, parasites and virus. You guys know what is the nosocomial infection?
Yes, ma'am.
What is that?
It's an
It is
infection.
yes, still. It is on the slide, but still.
Okay, when the patient goes to the hospital,
Mm-hmm.
in which the infection was not present, but during the course of treatment, it develops the treatment. It develops the infection.
Okay, so that is right when you are in the hospital, but not the cause of treatment. Stay in the hospital.
That's right.
During the stay in the hospital. In the treatment procedure, if something happens that is going to change in the physiology, that is a different issue. If there is a problem in the treatment. When you are staying in the hospital, if you develop the symptoms or any infection for which the patient didn't came in the hospital, that is called the nosocomial infection or hospital associated infection. What is the another name of the hospital associated infection? Now it doesn't say any nosocomial, it's calling another name. You know,
It's hospital acquired.
Acquired infection.
Yeah,
what is
hospital
it?
acquired infection.
Infection.
HAI.
HAI, hey.
Yeah.
Mhm. So nosocomial infection, associated infection also more difficult because of the high level of resistance to antibiotics. Patient develops symptoms of an infection with 40 hours. This is important. 48 hours after admission. The patient is considered to have acquired an HAI infection, hospital acquired infection. That is called the HAI. Nowadays, it's called the HAI, and it has to be 48 hours. Okay, within 48 hours after infection. What is opportunistic pathogen? Who, when a person is healthy, cannot do anything, stay there, transfer. When the patient or the person's immune system become weak for any disease, then they take advantage to overrule. So that is the opportunistic organism is an organism that does not usually cause an infectious disease in a person who has an intact or strong immune system, but can cause disease when the host immune system has been compromised by disease or another condition that has damaged or altered the immune status, like the HIV, cancer, diabetes, all these things. So for all those things, the immune system became little bit weakened. So who has the HIV? They doesn't die for the HIV virus. Their immune system becomes so weak they doesn't have any immunity. So every time they are getting sick, either diarrhea or cold or this and that, so ultimately, our body cannot take it anymore. So the group of saprophytic fungi, organisms that derive nutrients from living or dying organic matter, includes both filamentous molds and microscopic yeast. There, some yeast are also opportunistic pathogen. So serious infections in immunocompromised patients are often associated with pathogenic or opportunistic fungi. For pathogenic, obviously, they are going to take over the body who has the immunocompromised. But opportunistic fungi, if you have the strong immune system, they cannot show their action. They cannot show their pathogenicity. But who has the weak immune system, they can flourish and they can be infectious or show their pathogenicity. Infectious microorganisms can be present in the hospital or treatment facility and become a major problem for immunocompromised patients. Can anyone tell me which is the very common HAI?
Those immunocompromised because of HIV infection, ma'am.
Which is the common HAI?
Immunocompromised patients.
Which
Oh,
AAI can
elderlies, most probably. Sorry.
what?
Elderlies, ma'am. Because their immune
No.
system are
Either
quite.
name. I want the name of the disease, which is very often called in the hospital.
C. diff.
Oh,
It has to do with a respiratory
MRSA.
disease.
TB, most probably.
No, TB is the reverse
No.
isolation. Pneumonia.
Respiratory is
Pneumonia.
pneumonia.
Oh,
Yeah.
Pneumonia.
So
Oh, okay.
very common
Mhm.
HAI. Very
Mhm.
common HAI, especially the elderly person. Very common. Very common. People sometimes come with the different problems. Heart problems, this, that, whatever may problem. Ultimately, maybe they are getting cured from there, but they ultimately for long stay, lot of times they got the pneumonia.
Miss, what about like surgical sites infections, like after surgery, like a site gets infected? That's also common, right?
Mhm.
Or
It depends upon, that is not for the HAI. That is the how the operation has been taken care, disinfection,
oh, okay, okay,
sterilization, how they have been
sterilization.
handled.
Okay,
Exactly. That depends upon that one, how they are treating that one.
okay,
Okay. So
okay.
it's not
Thank
for
you.
the HAI. Yeah. That can be even you may not one day surgery. You are not staying in the hospital. Say for example, now they have day surgery, but
Yeah.
that surgery can be infected too. That is the total operating procedure, how they are doing it very in a sterilized way.
Okay. Okay.
Okay.
Thank you.
Okay, good. What is transient flora? Transient means the name is temporary. That transiently stay and can be gone also. Can include bacteria, fungi, and viruses, like the dandruffs. It sometimes comes, sometimes go, depending about your day skin. That's also infection caused by direct skin to skin contact or indirectly via objects. I think you understand that one. I don't have to clarify. They do not usually multiply on skin because our skin surface is not getting any nutrients to them. Amenable to removal by washing your hands. You wash. So most of the time, whatever in hand and we are, it is not deadly. Why we wash a hand? Always to remove the transient flora. And then you are going to sanitize your hand. So commensal bacteria, what are that? This is the relation between two different organisms that eat from the same dish. Neither benefits from the other or provokes harm. It is a neutral relationship. Like that, we have friendly bacteria that colonizes our body, like the gut. However, this relationship is more mutualistic, where they both obtain benefits. Like the Staphylococcus epidermis, it stays in skin and nasal cavity. Conibacterium, skin and nasal cavity. So Corynebacterium species is didn't tell the specific one. It can stay in the skin and nasal cavity. This is called the commensal bacteria, which a mutual agreement they are staying, mutual relationship. Parasites. That is called which infection which is caused by the parasites. Parasites are divided in couple of things, mainly protozoa, just the giardia. When you have the giardia, it's the giardia lambelia. You can see the here the generic name started with the capital letter. This is the species name. It is starting with the smaller lowercase, right? And all are in italics. There are some nematodes, that is another phylum, nematodes, that is the roundworm. This is the long worm which has the hook and which is hang in the our intestine. Roundworms. It's called the Enterobius vermicularis. We have trematodes. In the trematodes, we have the tapeworm, which is called the Tinea solium. We have another type, it's called cestodes. That's the fasciculo. Trematodes is fasciculosis. You doesn't need to remember. Just remember the tapeworm, remember the trematodes, remember the roundworms. That is fine. Protozoa, roundworms, trematodes, cestodes. These are also acts as a parasite in our body. Parasites doesn't help us anything, but they use our resources to multiply. So subgroup of parasites referred to as ectoparasites are invertebrate animals called arthropods, like the mites, lice, these are the insects. They stay in your body, got the irritation, and they try to eat from our body resources. And human crabs also are capable of causing irritation and infection. Other arthropods rarely cause diseases but serve as a vectors for bacterial or parasitic infection, like the mosquito. Cockroach. Cockroach moves from one contaminated food and one place to another place. Maybe some has some contaminated food. They move around, then they come, sit on your food, or and then it become infected. You doesn't know you are eating that food. Oh, it's so tasty, but you doesn't know
Yes,
what
ma'am.
is inside the taste. So
Yes,
other
ma'am.
pathogens. Yes.
Does scabies fall under parasite?
Who?
Scabies. So it's like it's bugs under your skin type thing. They're
Yes,
little tiny.
yes, yes,
Oh, okay.
yes, 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 yes. Other pathogenic infections, parasites. Parasites are organisms that live or in a host organism and gets its food from or at the expense of the host. So they are not beneficial. They are not working mutually. They are only taking everything from us, destroying the host body. So this is the different types of bugs. It's called the ectoparasites, lice, mice, scabies, everything. Helminths, which is the roundworm, some of the tapeworms, they stay inside your intestine or digestive system and soak everything and slowly block your total intestine due to their large size and multiplication. Protozoa, there are different types of protozoa. That is a cause the giardia. What is called another one? Oh, Plasmodium vivax, which cause the malaria. So these are the protozoa. Lyme disease. It is the tick-borne disease. It's called the Lyme disease. It is a spirochete. Its name is Borrelia burgdorferi. You doesn't have to remember the names. I'm telling you. This is a spirochete. Lyme disease is caused by a spirochete. What is spirochete? Is the helical and motile bacterium. You are going to see when you are going to see different types of bacteria. Is spirochete. It's a motile. It's like a, you have the DNA double helix. It looks like that one. That is called the helical structure. The spirochete is transmitted when tick Loxodus species feeds on a blood meal from a host. So tick bites you. The spirochete is in the blood. It then eat the blood from the blood meal of the host, and then is going to give infection to another one. So diagnosis of Lyme disease is based on the identification of the antibodies specific to the infective organism in the patient's serum or CSF. It can only be detected in the serum of CSF using the immunology techniques of the antigen antibody reaction. Protozoa are types of parasites found in environment. These are one-celled organisms, that means one cell that multiply in humans, usually transmitted by fecal or oral route. Can also be vector transmission like the mosquito for the malaria. Classification based on mode of movement, how they move, whether the flagella or the wing or the leg or the what is called legs, such as in chronic long-term infection, tissue damage is often due to an autoimmune response to the parasite and not host antigens. Ultimately, it goes to the attack the immune system. Tissue damage can also be due to the toxic protozoal production and mechanical damage, like the Lyme disease or skin or on the whatever is the infection. Disease or infections kind of include examples. Malaria infects the blood. What is the plasmodium? Toxoplasmosis is from the lymph nodes. It attacks the insect's lymph nodes, the eye and brain. And Giardia infects the gut. Our other pathogenic infection, viruses. Viral infections are very common. Virus cannot survive without a host, so it cannot survive outside a living host cell and must use a living cells to replicate and disseminate it into nearby cells. So invade one cell to another cell, multiply, invade another cell to another cell. Viruses are capable of infecting animals, plants, bacteria. Some common viruses are COVID-19, influenza A and B, norovirus, that is called the stomach flu, human papilloma virus, HPV, that is the worst, herpes simplex virus is called the cold sores, Varicella zoster is the tick virus. What is the chlamydia infection? It's a bacteria. It's common sexually transmitted infection is called the STI, is caused by the bacterium Chlamydia trachomatis. So remember, this Chlamydia is a bacteria. Okay. It can cause infection is more men and women, can cause permanent damage to female reproductive system. It cannot, if it is not cured properly, can cause potentially fatal ectopic pregnancy, can pass on to the baby if the woman is pregnant. It's so fatal. Often no symptoms are noted, but can be painful urination, white discharge. Specimen for detection include discharge, cervix swab, rectum, oropharynx, and conjunctiva swabs. Microbiology tests for diagnosis stained with Giemsa staining. Culture, more specific method of diagnosis, antigen detection, direct immunofluorescence staining, nucleic acid bed tests, rapid and inexpensive, but relatively insensitive in small numbers. So it's mainly you have to do the antibiotic therapy treatment. Rickettsia is also bacteria. Sorry. Sorry. These are the, you are going to later learn what is the gram positive and gram negative depending upon the cell structure of the bacterium. So these are the gram negative bacteria. Gram negative bacteria doesn't have the peptidoglycan layer that you are going to learn later. So gram bacteria, negative gram bacteria, these are called the ticks, lice, fleas, mites, seagulls, and mammals of the genus Rickettsia. So the rickettsia are a virus collection of obligatory intracellular gram-negative bacteria found in ticks. So in ticks, lice, flea, mites, they carry these bacteria and they infect human beings. Okay? And they can be found so in this. So these are okay. Bacteria are spread by the bite of an infected tick or mite. These zoonotopic pathogen cause infection that disseminated into the blood to many organs. It's spread from blood to different organs. In Canada, the only rickettsial disease observed to occur is Rocky Mountain spotted fever, caused by Rickettsia rickettsii, spread by the Rocky Mountain wood tick. So if you go to Rocky Mountain to visit Rocky Mountains, be very careful to take precautions from the bite against the wood tick. You are going, you are seeing everyone when the tick has been bite you or it's coming to your body or your clothes, and then ultimately it can make this problem. So it can be treated only with the antibiotics as this is a bacteria. Bacteria morphology. Now coming to the little bit more details of the size, shape of the bacteria. So there are three basic bacterial shapes. It is spherical, rod-shaped, and spiral or twisted. So one has spherical, one is rod-shaped, and one is spiral. This is called the spirochete. So how are they classified? The shapes of bacteria. This is, you can give the another picture of this one. So this is the round, this is the rod shaped, again this is the spiral. So cocky, these are called the cocky, these are called the rods, and this is called the spiralium. So here, this is cocky, this is the rod, and this is called the spiralium or spirochete. So different shapes of the bacteria. This is different shapes also. Now we are going to get little bit more differs. So this is, we call the, again, this is called the cocky. This is kind of rod-shaped. This is Trypanopallidium. This is the spiral. This is the Salmonella, is rod-shaped, but it has the tentacles. Vibrio cholerae, very dangerous. It has a tail. And it's rod-shaped, but with a tail. This is the Clostobacteria. This is the Clostridium. It's called the tetanus. Streptobacillus. This is the rod shaped, but in multiple. So one rod, two rod, three rod. We are going to come again later. Different of the arrangement. How it arranged. Diplococci. Cells remains in pairs after dividing. So when this stays in pair, multiply, 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 but this always stays in pair. This is called the diplococci. Single one is called the cocci. When it's only one and divide, 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 produce only one, 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 that's called the cocci. Diplococci cell remains in pairs after dividing. Streptococci cells remain in chains after dividing. Tetrad cells remain in groups of 4 and divide into two planes, longitudinal and vertical. Sarcine cells remain in groups of 8 and divide in 3 planes. And Staphylococcus is a cluster of bacteria and divide in multiple planes. Okay, so Bacillus, rod-shaped. Bacillus is rod-shaped. Diplobacilli Rod-shaped cells ribbon after dividing in duplicate. Streptobacilli chains. It's rod-shaped, but it divides and stays in chain. Palisades cells in a chain are arranged side by side, one after one, instead of end to end. It's not side by side. It's like you are putting the stack one by one, one by one. And Coccobacillus cells are short with a slight oval shape resembling both coccus and bacillus bacteria. You doesn't need to remember so many of this one. You have to remember what are the different shapes, rod shapes. This is Diplococcus, Streptococcus, Staphylococcus, very important, tetrad. These are going to come. Okay. So spirilla, what is spirilla? Spirilla is spiral shaped. These are elongated and spiral shaped. So these are called the spirilla. Is one of the three primary shapes of bacteria. Spirilla bacteria twisted and commonly occur in two forms. It's called spirillum when it's called the plural and spirochetes when it's singular. These cells resemble long twisted coils. Spirilla is plural. Bacteria are elongated, spiral shaped, rigid cells. These cells may also have flagella like you saw the previous one, which are long protrusions and used for movement at each end of the cell. So maybe at each of the cell they may have a long tentacle or it's called the flagella, which helps them to move very swift, very fast. An example of spirillum bacteria which is called the Spirillum minus, which causes the rat bite fever. Spirochetes, also spelled spirochet. These are bacteria long, tightly coiled. This is showing here, but is more tightly coiled. It's like a spiral. Tightly coiled spiral shaped cells. They are more flexible than Spirilla bacteria. This kind of spring-like action. Example of spirochete bacteria includes Borrelia burgdorferi, which causes Lyme disease. The Lyme disease, which is caused or carried by tick, is caused by the spirochete. So this you have to remember. The Lyme disease is caused by spirochetes. Okay. And trypanoma cause syphilis. That is also another spirochetes. Trypanoma. That is also important. Okay. So another very deadful spirilla is Vibrio bacteria, which is called the cholera also. They are coma stage. You see coma? So vibrio bacteria have a slight twist or curve, resemble the shape of a coma. They have a flagellum, which is used for movement. Number of Vibrio bacteria are pathogens and associated with food poisoning. One example is Vibrio cholerae, which cause the disease cholera. Okay. So identifying bacteria, how you are going to smear preparation and stain. You are going to make the smear, then you are making the slide and then stain, use microbiology. Identification of the bacteria involves morphologic examination under the microscope. How they looks like, whether they are coming single, pair, rod-shaped, spiral, cluster, with flagella, without flagella, everything. Cellular shape, arrangement and staining characteristics of the bacteria provide preliminary information as to the genus and species of the organism. Unstained bacteria placed on a glass soil and observed under the microscope will appear as transparent colorless structure and may be homologous or granular. So if you doesn't stain them, you cannot see anything. It looks like transparent. That's they are telling. Staining provides a means of visualization of and biochemical characterization based on the cell wall structure. So how the cell wall is made up of is a gram positive or gram negative. Then you can see if the cell wall is stained, you can understand the shape, you can understand the size, how it looks like, everything. Various staining process may be used depending on the suspected infectious agent and specimen under examination. So identify the genus and species of the microorganism and then determine the nomenclature rules are followed. Tell me, which one has been good? Which one is the right one? 1, 2, 3, 4, 5, 6, 7.
Which one is what, beneficial?
Nomenclature wise, which one is right? Which
Number
one is
one
right?
is good, ma'am. 1, 2, 4, 6.
Everybody is the same opinion?
Sure.
And
Whole
5.
class.
And 5.
Yes, Lactobacillus sp.
Lactobacillus, yes.
Whole class. Any other opinion?
Yeah.
Who is going? Who is
And
going
this
with? Who is
and the
going with
species
Luis?
has
Who
to
is going
start
with
with
Evelyn and Kara?
and the species has to start with the
Okay, let me
small
go to the
letter.
team.
What's the question,
Let me
ma'am?
get now.
didn't
Let
get
me
the
go to the
question.
team. Who is going with Luis? Raise your hand.
What's the
You
question? I
cannot
didn't
be
get
in. You cannot be in two groups.
I
Raise
don't
your hand.
understand.
Raise
I don't
your hand.
understand the question.
Sabana
What's,
is.
what's the question, ma'am?
The question is which nomenclature, as per nomenclature, which one is the right?
Correct. Yeah.
Okay, who is going with Luis now? Raise the hand.
Is right, as in, I don't get it.
Okay, so Kara is with Luis and Sabana, you raise the hand before. Luis, you raise your hand. Bharna, raise your hand. Who wants to be with Luis? Okay, Luis, no, just in the. So 1, 2, 4, and next page. Let me see the first page.
No.
Okay, Maris, Santiago, then Kara is already done. So you are only with Luis. That's the number 2 is right. And okay, down your hands. Down your hands, please. Maris, down your hand. Maris.
Maurice.
I'm done.
Maris, down
I'm
your
done.
hand. Maris, are you there?
Wait.
Okay. Again, you put your hand. Okay, now it's clear. So can I guess? Can I think that only 7 people are with Luis and all others with Evelyn, and Kara also was in Evelyn's group. So right now she is now Luis' group. So now it's
Yeah.
only Evelyn.
I
Rest
didn't
of the team is with Evelyn.
I don't even know the question. So I just stepped out to get a cup of water.
I told you the question again. What then?
You said which is right? Right
Which
in
one
what?
is
I
the
don't know
right?
what.
Which one is the right?
I cannot say what is the one that is the right. I can't see the options. I'm seeing your screen. I'm seeing our pictures.
I showed you before, so that's
Yeah, I
why.
was not there. I went to get a cup of water.
Okay. Here is the.
Okay. Staphylococcus. Staphylococcus aureus seems right.
I want to go on my own.
Staphylococcus aureus.
And Kara wants to go to your own? What
Yep,
is your?
1, 2, and 4.
You want to go 1, 2, or 4?
1, 2, and 4.
No, Louis, you cannot change. You want to change all decisions? Why?
Louis, you're in it already to win it, bud.
Isn't it?
Yes.
Yeah, Staphylococcus is not italics.
Well, in that case,
So 2, 4.
It would be number 2 because it follows all the rules. No.
Number 2.
So then it's just number 2.
So and
2,
4.
4, and 5.
2, 4, and
Yes.
5.
5? Why is
It
No,
cannot
but
be
5. The
five.
second one
Because
is
the
capital.
species
though.
No, it's not 5,
The
guys.
species is
5 is
in
not
the
supposed to be capitalized in the
uppercase.
second
It's not
word.
supposed to be capital. If it only.
The capital letter is wrong in the second word for number 5.
Yeah.
I'm getting anxiety.
I'll go with 2
So 2 and 4. 2
Don't
and 4.
don't be Kara.
I
You
think
can
it's 2
always
and 4.
Imam can always correct it. But for me, I will stick with 1, 2, 4, and 6.
2 and 4. Number 1, yeah, because the point of the italics. Number 1 would have been correct for me, but without the italics, it seems
Yeah.
maybe now that is wrong because it's not in italics.
Imam
So
can always correct
majority of the class is telling 1, 2, 4, and 6.
Yes, ma'am. The Lactobacillus species.
The
Okay,
right
so
one
then-
is only number 2.
Number 2, right?
I
Oh.
agree with you, ma'am. Thank you.
Wow. Okay.
Wow.
Yeah.
Even
it-
if the SPP has to be in italics.
Okay, ma'am. Thank you.
So everything needs
Yes.
to be italic and small.
First one should be started with the capital letter, genetics name, lowercase, species
Yeah.
name, and it has all to be italics. Then you change your mind. That means you are not confident.
Exactly.
Don't shout. If you are not confident, you change your mind.
Yeah.
No. Okay, so quiz setup. That is a quiz. 8 points. Oh my God. How am I going to set the quiz? Okay, due date is today. You can do that today. How to do that? How I'm going to set the date? Not in grade book.
Yeah, I think this is just a practice, ma'am.
It's just a practice,
Like,
ma'am.
because
But
it's
did get the
unlimited
8 points? I
attempt.
don't know. Okay.
No,
Okay.
it's not for marks.
Yeah,
It's
yes,
not
ma'am.
for
It's
marks?
unlimited.
Okay.
Yes, ma'am.
No,
You
it's
even know.
unlimited
Okay, so
practice.
you guys want to do it?
Yes, ma'am. It's
Okay.
unlimited.
Okay.
Yeah.
Okay. What is this?
Oh, they're too tiny, ma'am. They look
I
like
cannot
a
do anything. I
No.
cannot zoom it, dear.
Oh, that's
There is
cool.
no
Yeah.
options for the zoom.
Oh, okay.
Yeah, they look like a cockeye to me
Okay.
also.
To everyone, it looks like a cockeye. Everyone looks like a
Okay.
cockeye.
Yes, ma'am.
Okay.
Yeah.
Okay.
Yeah.
It's also cockeye. Cockeye in change.
Okay, engines.
Okay, and jeans. Okay.
Spiral. Spirited.
Spiral.
Spirillum.
Spiral.
Spirillum.
Sperilum.
Spirillum.
It's
Rods.
Right.
Rods.
rods.
Bacilli.
Bacilli.
Bacilli, everyone?
Yes.
Yeah.
Yes.
Yes.
Diplococcus.
Diplo.
Diplococci.
Diplococci.
Cocci. Diplococci.
It's mixed, right?
Yeah.
Cocaine. Yeah, exactly.
It says select. It says select all that apply.
All that.
Read the question.
Yeah.
So
So
cocci
Cocaine.
and
Right.
bacillus, I think. Bacillus.
Bacillus.
Okay. Next one.
That's also bacillus. Yeah. Oh no, spirochete. I think. Spirochete.
Spirochete.
Okay. So I'm clicking the spirochete for you guys. For me is Vibrio. Okay. We'll see whose answer
Yes,
is
ma'am.
right. Okay.
Yeah.
Okay.
It's also
Tetrad.
Okay. Submit quiz. Submit quiz.
Oh.
What is that?
Oh.
Which one?
Let's see.
Who is right? Who is wrong? Let me see.
No,
I win. I win.
Oh,
Mom
I
is
win.
right.
I win.
you
I
won.
win.
What about the
I
last
You
one?
win, mom. You win.
That's the last one. Okay.
Oh,
So see,
tetrad
I didn't tell you.
bacilli.
Reply. You have tetrad and you
yeah,
have the
you have
chain.
a
So it
club
is
head.
in yeah chains. You didn't tell the chains. Okay. Chains of
Yeah, okay,
cocci.
ma'am.
Okay. Done.
Mm-hmm.
I'm going to go back. Oh dear. I'm going to go back now. Oh, back to manage quizzes. No. Discard your changes. Yes. And then where I am now? Dear Jesus. Content. Okay. You done? Bacteria morphology. So you're done for the unit number 1, I guess. So let us start unit number 2. What is unit number 2? Microscope. Sterilization. I'm not going to go through this one. Just you guys read it. This is already done so many times. I told you guys. Protection laboratory personnel provides person must be followed for the rule of general safety. Protection, you guys need that. Pathogens are processed, cultivated, analyzed. You guys know this one. Risk group already told. Oh dear. Sterilization technique. What are the different sterilization techniques? Can you tell me? Sterilization, you can be done by what? Autoclave and
Autoclave.
Hot
Dry heat
hot air
oven.
oven.
hot air oven and
The loop.
no. Which cannot be autoclave or which cannot be go for the what is
Yeah.
called hot air oven, like the stethoscope, the plastic or the polythene things. What is going to use?
Dry.
No.
Some
Oh, bleach or glutaride or whatever the hell it's called.
No. Bleach cannot sterilize things.
Miss, wait, what's the question? Sorry.
Is it radiation? I can't remember.
How you can sterilize things which cannot use the autoclave or the dry heat oven or hot water?
UV.
No.
Ma'am, is it radiation?
No.
What is it?
This is the problem. Not to review whatever you have studied. You can see everything comes. It's a cycle. You forgot about the gas sterilization?
Yeah, gas. Yes, that's right. I knew it was a form of
yes, ma'am.
matter
Yes.
that is different from liquid. Yeah, gas.
Actually, I googled,
That's why I
ma'am. I
was.
forgot also.
You
I
Google
don't know
it.
my
Yes.
Yes,
radiation
ethylene
chemistry.
Yes.
oxide.
Yes. But I cannot remember. I have to Google
Yeah,
it.
Yeah,
And being over 60, I have to remember that.
it's
I
gas.
forgot to, ma'am. I forgot. Yeah, the ethylene oxide.
Exactly,
Decomp-
ethylene oxide.
instead
Ethylene
of
oxide.
gas.
Yeah. How it kills it?
Ethylene oxide.
Because of
Don't
the
Google.
chemical
Don't
transformation.
Google how it kills the pathogens. Which level?
Oxidization.
What?
It
As
oxidizes them.
at the name is ethylene oxide, so it's oxidization.
Yeah.
Yeah.
Yeah. How it calls?
So what
What
do you mean by
oxidize?
level?
What it oxide? What it oxide? If it is true the ethylene oxide is getting oxidizing, what is oxidizing? What is your thinking?
Both the spores and the cell wall.
Well,
It
the spores of the
desiccates them.
bacteria most probably, ma'am.
If the cell wall till the bacteria can survive,
Yes, the
Yeah, it
spores,
because
ma'am.
desiccates it, like
Bacterial spores.
evaporates it completely to non-existence.
it damage, destroy the DNA of the pathogen.
Okay.
Okay, which is the hereditary material for the any pathogen, which is the bacteria or anything. It damage the DNA, it chopped, it destroyed the DNA. So the whole thing is dead. It cannot multiply. What is the antisepsis means? Where you do the antiseptic? What is the common antiseptic you use in our daily practice?
70% alcohol.
70%
And
alcohol,
yeah,
70%
it's used for
sulfur.
humans, ma'am.
No,
For
not only for humans, for who vets also use that.
Yeah, to reduce
When
the
they do the
germ.
surgery, what is the and where it
Yeah.
used?
On the
skin.
surface of the skin.
In the
Skin.
surface of the skin.
Yeah.
Yeah, to reduce the number of microorganisms in that area. So antisepsis is the process used to decrease. It is not sterilization. It is not causing the totally your micro pathogen or any microorganism free. It is process used to decrease the number of microorganisms that are present on the skin. It didn't say here pathogen, any microorganism. It may be some pathogen, may not be normal. So it is the more, and you doesn't know which normal microorganism can be pathogen to something else. So it is the process to use to decrease the number of microorganisms that are present in the skin. Antisepsis is on the skin only. Typical antiseptic agents: alcohol, 70% isopropyl alcohol, chlorhexidine products. When you use the chlorhexidine, which must we have to use the chlorhexidine to use as an antiseptic?
In surgeries, ma'am, and also when we collect for alcohol, blood alcohol.
Blood culture.
Yeah.
Blood
Blood
culture,
culture.
that's must, and blood alcohol. Yes, yes. You can also betadine, iodine, providine, iodine, but that gives some color. But chlorhexidine doesn't make any color, so now it is most popularly used the chlorhexidine. So alcohol is often used to disinfect the skin before drawing blood or vein puncture. Disinfection also destroying the pathogen organism, but not their spores. Disinfection means you are destroying only the pathogens, not their spores. Which can destroy the spores? Which procedure?
Autoclave.
Sterilization.
Sterilization.
Not only
Moist heat
autoclave, the dry heat oven, the ethylene oxide gas, all can. So it is the
can.
sterilization procedure. There
Yeah.
are three different types of sterilization. What is steam sterilization? It is the hot air oven sterilization, and is gas sterilization. Mostly common is the sterilization through the autoclave. But there are the where you cannot use autoclave, that time you have to use either hot air oven or the ethylene oxide gas, depending upon what you are sterilizing. So disinfection is the process of destroying pathogenic organism, but not necessarily or microorganism or bacterial spores. Physical, physical moisture, dry heat, or chemicals like the bleach, phenol, alcohols, aldehydes may be used. sterilization, destruction of all microorganisms, including bacterial spores. What are the indicators we use to make sure the all in the autoclave to make sure all spore has been destroyed?
Biological indicator.
Biological.
Somebody is listening to the course and talking to some other person.
I think that was Santiago.
Santiago is singing or talking to other
No,
people?
miss, I had my mic off the whole time. That was not me.
Oh.
So who is talking? It's like a male voice. Either you or Luis or someone's.
Miss, you
Very
can play back the teams and you can see it's not me. I'm not sure who it was.
Sorry, miss, maybe it was my husband in the background.
Oh, okay.
I turned off
Somebody.
my mic.
Okay, no problem. Yeah. He's really telling to make food for you, him?
No, miss, he's on the phone. Sorry.
Yeah, I'm just joking. Yeah, so sterilization. No, if you are making the food, maybe I can start and go to your house to eat. I'm joking.
Anytime, miss.
I'm just joking. You are already overtired with the course, taking care of the kids. Don't accept anyone now, later. Sterilization refers to killing or destruction for all microorganisms, including bacterial spores. Sterilization may be achieved in various ways, as I discussed, generally involving physical means such as heat or filtration and chemical means such as oxidation. That is the chemical way. Use of disinfectant in microbiology laboratories. What is disinfectant you use? 1% bleach. How you do that? 1 in 10. So 1 part of bleach and 9 parts of water of household bleach. What is that? What is the percentage? What is the strength?
1%.
1%.
No. What is the original stock solution?
100.
95.
No.
What?
How the bleach come?
95.
How
The
the
bleach
bleach
comes in a
come?
bottle.
10%.
I am not going to tell you. Next, you are going to go in the class, look at the bottle, or you go and look at your bleach if you have something in home, and then you are going to tell me tomorrow. Okay? So is 1:10 dilution, but tomorrow you guys have to tell me what is the strength of the household bleach. It can be used for spill control, can be used for wipe down of surfaces and equipment. Lysol is a phenol. We use the Lysol, but this is originally biochemical name is phenol. So if using alcohols as disinfectant, care must be taken so that alcohol does not evaporate prior to the required contact time. So that's why you are not using absolute or 100%. Alcohol never comes in 100%, it comes in 95%. That is equal to 100%. So 90%, 95% alcohol is more strength, but it cannot disinfect properly. Why? It evaporates very quickly. It doesn't get time to touch the microorganism for some more time to distract them. That's why the 70% alcohol is used as a disinfectant, because it stays some time on the surface to interact with the microorganisms to destroy them. Is that clear why we use the 70% alcohol? We did it before. Heater burning. Heat is widely used and efficient physical means of sterilization. Heat may be employed in the form of dry heat or moist heat. So moist heat is autoclave, dry heat is the dry heat oven. Dry heat destroys bacteria by oxidation, and moist is destroyed organism by denaturing potency. You doesn't need to. If you can remember, fine. If not, it's okay. The type of sterilization method is used will depend on the nature of material being treated. That is important. Dry heat carried out in a hot air oven. Temperature for the dry, this is important. Dry heat Sterilization needs 160 degrees centigrade for minimum at least an hour. What is the temperature for the autoclave?
One,
Come on.
121 degree.
121.
121 degrees.
1 degree?
Yeah.
No,
121.
121
Of what?
Celsius.
degrees
Celsius.
Celsius.
Celsius for 15
And
minutes.
what is the working principle of the autoclave?
What?
Working principle or sterilization principles of the autoclave.
Pressure.
Moist heat.
Oh yeah, pressure.
Yes.
No?
Moist heat.
It's like heat, right? Is it heat?
Moist heat.
Steam pressure.
You need to wait to achieve 121 Celsius to
15
start the
PSI.
autoclave.
Is it 15 psi?
Sorry, ma'am, I also forgot the PSI, but it's usually for
It's
15
15.
minutes,
It's
121 degrees
15
Celsius.
PSI.
But the PSI,
No,
It's
15
15
pressure,
PSI.
I forgot.
So what is the
121
working
15
principle?
PSI.
What is the working principle?
Sterilization.
You wait until that's
You need, yes,
Sterilization is the
ma'am, you need to
procedure.
wait.
pressure.
You need to
Sterilization
wait.
is the
Moist heat under pressure.
Ma'am, you need
heat
to wait.
under pressure. Heat
Yeah,
under
heat
pressure.
under
Moist
pressure.
heat
No,
under
not heat, not
pressure.
heat. Sorry. Steam
It's
under
moist heat.
pressure. Steam
Oh,
under
Steam.
pressure. Steam
Oh,
under
okay.
pressure.
not moist
Okay,
heat.
Steam
miss.
under pressure. Okay? And it's minimum 15 degree, 15 minutes.
Minutes.
But it
15
also
minutes.
Yeah.
depends at the size or the load size. If you are
Right.
putting lot of things, you may have to increase the time from 15
Yeah.
to 30, sometimes 1 hour even, depending upon what the load or the size you are getting sterilized. Minimum is 121 degrees Celsius, 15 psi, and minimum 15 minutes.
Okay, thanks, ma'am.
Okay. If the temperature is decreased, the time must be increased. That is the proportional. So if kind of you see the temperature decreasing, you have to put instead of 1 hour, maybe 1 hour, 1 hour half, 1 hour half. So what you can use so that it doesn't break or crack in the hot wire oven and also in the autoclave also. Only the borosilicate, you remember, heat resistant, the glass material.
Yes, ma'am. Glassware.
Remember the borosilicate? Borosilicate is the heat resistance. Only this can go in the dry, hot sterilization procedure. It has a high thermal resistance that is commonly used to make high quality labware, and this product can be sterilized in hot air oven. So hot air oven can sterilize equipment that destroy microorganisms, including spore-forming bacteria, requires a temperature of 160 degrees Celsius to be destroyed. Non-sporing can be killed at 100 degrees if there is non-sporing, but for sterilization method is the killing the spore-forming bacteria. So if it is 100 degree temperature, you are running everything in the 100 degree, it can be disinfect or it can kill the it cannot kill the spores. So it is not going to be sterilized. It will be only disinfected. So if you try to disinfect or sterilize anything at 100 degree in hot one event, is it going to be sterilized? No. For sterilization, you need 160 degree temperature. All items to be sterilized are wrapped and loaded into the oven in a way to allow heat to contact all surfaces, so that you have to put them in loose and separate. When the temperature of 160 degree is reached, the timer is set for 1 hour. Then when you are done, allow oven to cool before opening the door. Entire may take 2 to 3 hours to complete. Hot air oven is unsuitable to sterilize rubber goods or equipments containing rubber, washer, stoppers, equipment made with both glass and metal. Can you tell me why? if any instrument is made up of metal and glass parts, why you cannot use the hot water oven?
It will spark.
What?
It will dull. It will
Yeah.
dull, ma'am. The instrument.
No, we are putting the instrument separately. You can do that. You see, one instrument which is made up of metal and glass, both can go in the dry hot oven separately. But when it's made up of metal and glass, why you cannot put in the hot air oven? Anyone?
No.
Yeah. Or like
Your principle is right, expansion. The expansion capacity for the glass and the metal is different. Metal expands more faster than the glass. So what is going to happen is going to break, because metal is going to make more expansion in comparison to the glass. The things is going to break. Okay. Yes, you always do that. You always do that. Only cannot, at least I appreciate you. At least, no. At least I really appreciate you bring the expansion thing, physics, the expansion things. But you didn't able to finalize it. So you make the basic structures and I just put the cream on that. Okay. So hot air oven is unsuitable to sterilize rubber goods or equipments containing rubber, washer, udder and metal and glass because of the unequal expansion of either materials, or the melting or the fixative holding of the equipment. So there is the fixative or something there, then which is jointing in the joints. It also cannot sterilize liquid chemicals, as most are flammable, combustible, noxious, and also if you put that one, it's going to dry. You are going to make one concentration solution and put in that, even it is non-flammable. You cannot put that one there because of the is going to evaporate and dry, and the concentration is going to change. So do not put sealed containers in a hot water oven, as the containers will expand and explode. Everything should be open. Hot air oven is identical to sterilized substances that are insoluble in water, the gels, lubricants, or dry chemicals and powders. It also sterilizes metal instruments with sharp cutting edges like surgical scissors, scalpel, blades. Because if you put them in autoclave, we are doing it, but the autoclave, it can be blunt or rusted. Moist heat will dull the blades. That's why dry heat does not. To clean the hot air oven, unplug and wash the interior with lap soap and water or dilute bicarbonate solutions, 2 to 3 grams per liter. Sport a thick walled protective capsule produced by some bacteria highly resistant to heat and chemicals. Sterilization by moist heat. One of the employing moist is boiling water. You can do it. Boiling water can kill the vegetative forms of bacteria but not their spores. So certain species of bacteria, including genus Bacillus, have the ability to form spores under unfavorable condition but return to the vegetative or active state when favorable condition return. So that is when they doesn't have anything, they will sit like a spy, will see, and when the favorable condition will come, they are going to, you are maybe already infected with that, but it's not getting your favorable condition. So that's why it's not applied. Whenever that is the opportunities, opportunistic. So whenever the favorable condition will come, they are going to explode. Spores are highly resistant forms of bacteria and thus pose a problem in sterilization. The most effective means of sterilization with moist heat involves steam under pressure. Okay, this is the working principle using a special device called an autoclave. If the method of choice any material that is not damaged by moisture, high temperature or high pressure, most of types of media. What if the cultural media are used in the microbiology are sterilized in the autoclave? What is the media? The media where you grow the bacteria. That is called the media. It's called the food for the bacteria or the favorable conditions for the bacteria. Sterilized in the autoclave. Contaminated materials or used media will also be sterilizing using an autoclave, then discarded. Even whatever you use, the bacteria or anything or culture, you have to sterilize it so that it is not going to spread the infection. Autoclave provides sterilization by moist heat under pressure. It is the most reliable and effective way to destroy microorganisms and spore-forming bacteria. Autoclave uses the principles that water use pressure. So you are going to water, you are going to give the temperature is rising, ultimately is making the pressure high pressure, and then with the high pressure the water is boiling and high temperature, then free flowing steam. So technical aspects of autoclaving are the best learned by practice. So strict attention should be paid to the operating manual. You guys did the autoclave, right?
No.
With Saina, you didn't do the autoclave?
No, we got like a brief description and shown like the paper and the biological little thing you put inside, but we didn't ever ran it.
Okay, so I have to tell Saina to do that. The following is the general guideline. Operation: before use, clean out any dirt which is collected at the bottom of the autoclave. Always you can use the distilled water in the reservoir when it is required. Place articles to be sterilized into the chamber. Articles for terminal disposal are put in the autoclave bags. You have the autoclave bags which has going to we are going to discard. Terminal disposal means you are used already. They have the contamination of bacteria or virus or anything, whatever it may be, mainly the bacteria. So you are going to put in the autoclave bag, and these autoclave bag can resist the temperature and the pressure and the heat. So then you are going to put it so that after sterilization, you can dispose in the biohazard bags. So article to be sterilized and stored are wrapped in muslin. You guys see what is the muslin paper or autoclave paper. There are autoclave bags. You have seen that the bag, you have already seen that one, bags where you can put it. And then you have the autoclave tapes, which autoclave tapes looks like white, but after autoclave, what is comes out? How it
Black
looks like?
stripes.
Stripe,
Black.
black stripe, yes. The what it does, putting this one, it helps to
Indicate.
stop. That is my alarm, 1 a.m., 1 p.m. So yeah, sorry. So then you are going to wrap it in muslin, place in properly with a gap in between. So only the plastic, that is the important. Not all plastic can be autoclave because it cannot take the heat and pressure and steam. Only plastic can be autoclave is polypropylene. Okay, this has to remember. Okay. So autoclave, you guys know, is 121 degrees centigrade. Set the temperature 15 minutes or more. Pressure is 15 pound psi. Okay. Vent the autoclave to release pressure before opening door by turning to the dial. So when you put the pressure, close door, so you feel everything, close the door, removes, then you run it. When it started to get the temperature, then it's already set the 15 psi and 121 degrees centigrade, then you are setting the temperature for 15 minutes or more, not from the starting. When you put the on, set the 15 minutes, leave, no, you have to watch. When it's starting, reach the 121 degrees centigrade and 15 psi, then you are setting the timer. And then vent the autoclave to release pressure before opening. When it's done, so you have to put that vent so that the inside pressure, what is there, is going to release out. Otherwise, you cannot open it. If you open it in this, you cannot open it. In case it opens, it's going to severe burns. Autoclave is a heavy metal chamber with a door or lid that can be fastened with withstand internal steam, pressure, and has a pressure gauge. It has a pressure gauge to show you the pressure. This is, exposed to steam at 120 degrees, 121 degrees centigrade, 15 minutes. PSI, that is the 15 pound per inch. This condition will kill all forms of bacterial life, including spores. Temperature chart recorders must be used to documentation of the autoclave maintenance and quality assurance programs. So steam autoclaves are designed to use high pressure and high temperature steam to kill microorganisms. They're also used to render biohazard materials inactive. So you guys know that. So this is the conversion. Temperature 121 degrees centigrade as per CSML's lab safety guidelines. Pressure 15 psi, or in terms of, generally this has been used 15 psi. Okay, but it is equal to 103.4 K Pascal. Time 15 minutes or more. Sterilization involves absolute destruction of microorganisms. You guys know, purines, viruses, everything. Okay, absolute destructing means just that. So nothing, no pathogen or no any forms of the pathogen is going to be there. There is no such things as partial sterilization. Partial sterilization means disinfection. It's not sterilization at all. To achieve a sterilized state can be difficult as well as be difficult to prove. Under in circumstances, steam sterilization is the preferred method for sterilization and works by denaturing proteins. The steam sterilization works by denaturing the proteins. Hot dry oven by oxidation.
I missed to interrupt you. It's 103. Just wanted to let you know.
I know. My alarm rings at 1.
Okay.
But till I put in snooze, so don't worry. So you guys know this one about this how autoclave works, right? Increase temperature, pressure, everything, killing ability is the same thing. It's going to repeat and repeat and repeat. This is the structure. Okay, this is the structure. Items that can be autoclaved are cultures and stock of infectious materials, culture dishes. Okay, so this is the another question. Can you autoclave the live vaccines? No, because it's going to kill. Discarded live and attenuated vaccine, contaminated solid items such as petri dish, appendoptis, everything, items for sterilization such as glassware media is 105 now, not 13. Okay, items that cannot be autoclaved, snooze. Give me couple of minutes. Materials like the solvents, volatile things, chlorinated compounds, it cannot be. With it, chemotherapeutic agents, radioactive material, some plastics, you cannot autoclave without approval or the autoclaves which is only designated for those. Okay. These are the, we are going to from the tapes tomorrow, but I think you guys all know what is the tape looks like, what is the sterilization indicator, what is the chemical indicators. You know that, right? The chemical indicators. Do you have to go through that? Biological indicators, physical, you guys know that, right? Yes or no?
Yes, ma'am.
Yes,
We know.
ma'am.
We know.
Yes.
Yes,
Okay, so
miss.
okay. So we can go from tomorrow from this slide number. What is the slide number? This one, 40. Okay.
Okay.
Preparation material for autoclaving. Okay. Okay. Go.
Okay, ma'am. Thank you.
Thanks,
Sorry for
ma'am.
taking extra 6
Thank
minute times.
you.
I know because it rings on me. But sometimes when I starting things, I cannot complete exactly. It's already the repetition. Whole class today is just the repetition. Okay. Bye.
It's okay, miss. Thank you.
Okay. Bye.
Yes,
Bye,
thank
ma'am.
you.
Thank
Thank you,
Okay,
you.
ma'am.
ma'am. Thank you. Bye, everyone.
Bye.
Bye.
Bye.